That's Leandro Xavier Neves from the University of Liverpool.
Hello, my name is Leandro. I'm a postdoc in Nemo's lab in Liverpool, and I will briefly talk about the poster I'm presenting today. It's titled Insights on Regulated Proteolysis During Coronavirus Replication. It's poster number 2. So if you're interested, please come have a look and discuss. In the past years in the lab, I've been basically studying proteolysis. We use proteomic techniques to study proteolysis in different viruses, especially coronavirus replication. And we all know that proteolysis it's an important mechanism for viral replication because this is essential as the viral proteases, they mediate the autoproteolytic processing of the polyprotein, releasing the non-structural proteins that will form the replication complex. And this replication complex will be responsible by synthesis of negative strand and positive strand subgenomic RNAs. It's interesting that the same complex will have the ability to produce both strands, and let's call that there is a conversion step that actually regulates the configuration of this complex that defines which strand it's being produced. And we don't fully understand what governs that conversion, but we have current hypothesis that the extent of the proteolytic processing of the non-structural proteins is behind and what part of what regulates the two configurations of the replication transcriptase complex. To study that, we, with help of Max, actually has set up using reverse genetics a system in the lab that we have a MHV mutant that has a temperature sensitive NSP5. So when that virus is replicating at 39 Celsius degree, NSP5 loses its ability to cleave the polyprotein, and we can basically interfere with the polyprotein processing at specific moments of the replication cycle. And we use this. This is actually our setup. We've got cells infected with recombinant MHV virus and also the temperature sensitive mutant. And we kept the cells, some cases at up to 12 hours in 33 Celsius degree, but in two conditions we had the temperature shift to 39 to block the proteolytic process. Early stages at 4 hours or 8 hours post-infection. And then we look at the impact of that temperature shift at protein levels using proteomics. So these box plots, they represent this as an example, the nucleoprotein and the spike glycoproteins and polyproteins. But for all accessory proteins and structural proteins, we see the same pattern, which basically shows in red at 4 hours when the temperature shift happens at 4 hours after infection. We have a depletion of viral proteins in the system, and that suggests that we don't have replicase transcriptase complex that has been converted to the production of positive strand RNA. But in later time points, we don't see that effect anymore, showing that the conversion has already happened at that point. And also by looking at using proteomics to look at cleavage sites along the polyprotein, we can see that in both cases, irrespective of where we change the temperature to 39 and block the proteolytic processing, we see an absence of cleavage in the polyprotein, but together those results show that the late change in the temperature is not affecting the replicase transcriptase complex anymore. Yeah, with that, I would like to thank you, the lab members. Thank you all for your attention, and if you have any questions, please come.